For our last session of the day, it is my privilege to share the session with two postdoctoral fellows. The first is Dr. Emily Mankin. She did her PhD at UCSD with Jill and Stefan Lukeb, publishing in high-impact journals such as Neuron and PNAS, and came to us at UCLA and is doing a postdoc with Itzhak Fried in collaboration with myself. She has been awarded the prestigious Giannini Fellowship for Postdoctoral Fellows and has already been published in another high-impact journal in eLife, which she will talk to us about today. All right, thank you.
Thanks. So today I'm going to talk about our recent work on using microstimulation to affect human memory performance. But I wanted to start with just a really brief introduction to this whole session, which is called Neural Implants in Clinical Populations. And so I know you already know this because this is the end of the day, but the general broad strokes goal of neuroscience is to understand the brain and also to affect or treat the brain. And if we're being honest, for I think the majority of neuroscientists, we can throw the word human in there, that we can use other model systems, but most neuroscientists, I think, are really, truly, deeply interested in the human brain. But largely, we tend to study animals because by working with animals, we have the ability to observe and manipulate at these very small scales, the microcircuit level, the single cell level, and even smaller. You can do very specific things through optogenetics. You can pick specific cells, specific populations, etc. Human studies, on the other hand, have the advantage of that you're studying humans, but your ability to observe and manipulate tends to be limited to the things you can do non-invasively, which tend to be at that like macrocircuit brain region and higher level. But the ability to do neural implants in clinical populations helps us bridge that gap. Because neurosurgical patients sometimes undergo invasive procedures for their own clinical purposes, for their own health benefits. And so that gets around the ethical issues of implanting electrodes into people's brains, allowing us to get some of those advantages that you have in animal studies, but studying humans. And so there's a growing number of neurosurgeons who are interested in using their clinical patients for research to be able to study the human brain at these fine spatial and temporal resolutions. And so all of us in this session are going to be talking about that particular instance. With Dr. Fried, we tend to work with patients with epilepsy. And so the way our setup typically goes is that these patients with epilepsy will have electrodes implanted throughout their brain in the regions that neurologists suspect could have seizure-genic properties. And they're being recorded 24 hours a day in order to capture their seizures. But in the meantime, while they're waiting in the hospital, we can come in and ask them if they'd be willing to play some games, do some tests, etc. And we piggyback on their electrodes to be able to record. And so you can see, so here we have these large clinical macro electrodes that pick up large areas of the brain, and then we enhance these with micro wires, which are about 40 micron in diameter, and allow us to pick up the activity of individual neurons. And so here are some example neurons one could record. And so specifically in our lab, we are interested in a number of things, but especially memory, because our ability to make and maintain memories is certainly a very fragile ability. There are lots of conditions in neurologic diseases that can lead us to lose that ability, and it's having a growing impact as our populations live longer. And so being able to treat or affect memory would be really significant. And right now there are really no satisfactory memory restoration treatments. But it's worth noting that brain stimulation is something that has been used in the past for successful work. You've heard about some of these today. Many of them have even received FDA approval. And the four that are highlighted in yellow there are ones that have actually been done with intracranial electrodes. The others have been non-invasive. And then there are a wide variety of other conditions that people are continuing to explore that haven't yet gotten approval. And so we are looking at this last one of memory disorders. And Nantya Suthanad published in 2012 pioneering work on using entorhinal stimulation to improve spatial memory, where they showed that if you learned the location of a place in a town, in a virtual town, while you were receiving stimulation to the entorhinal area, when later you attempted to relocate that place, you could go there more quickly and more directly for the items that were stimulated than the items that did not receive stimulation. But it's worth noting that stimulation was applied through these large clinical macro contacts. And there are several other labs that have been doing similar work using stimulation through these large macro. They do bipolar stimulation, so it goes from between these two large things. But if you look at the MRI and where those actually end up, you can see that they end up kind of impacting several different brain regions, and they cover a really large portion of brain tissue. And so we wondered whether it might be possible to do something a little more specific, a little more on the scale that animal studies have used by using a microelectrode. And unfortunately, I don't have a photo of our actual microelectrode, but essentially in place of one of these 40 micron wires, we put a 100 micron stimulating electrode. And so it's quite small and is really very focal in terms of where we're able to target. And our goal is to target essentially the perforant pathway, which is the pathway between the entorhinal cortex and the C3 area of the hippocampus, since the hippocampus sits at the very top of the memory processing hierarchy. This also allows us to mimic some animal studies that have used microstimulation of the perforant pathway to generate long-term potentiation, acetylcholine release, and hippocampal neurogenesis, all of which are very memory related. So we targeted that. And it's worth noting this paper has 3 co-first authors because this paper really existed in 3 stages. Michael Hill was very involved in developing the idea of microstimulation. He did a lot of the safety work and hardware development. Ali Tatiz was very involved in collecting the data and getting patients to do the task. And then I came in along with Sarah, who's going to speak in a minute, and we did a lot of the data analysis and got the paper to where we really had some interesting results. So in the task, we asked people to do a person recognition task where they looked at novel people, and on half of the trials, we provided a theta burst stimulation protocol just before image onset, and later we asked them to discriminate whether they had seen photos before. And when we analyzed, we looked at the mean change in remembered rate. So essentially a positive number here indicates that performance was better on stimulated than non-stimulated trials. And we divided our analysis up into where the stimulation electrode was located, and we divided it both between left and right hemisphere because it's been suggested that hippocampal processing is lateralized in humans, and that faces and visual stimuli tend to be more processed in the right hippocampus. And then also between whether the stimulating electrode was in gray matter or in the angular bundle, which is where the perforant path resides, since we thought that perforant path targeting was really our goal. And what we find is that, in fact, if you look exclusively at the change in performance when you have the stimulating electrode in the right angular bundle, which is where we would have predicted our highest effects would be, we find an almost 20% increase in performance for stimulated versus non-stimulated trials. And so this is really exciting because it shows that microstimulation with a very small and probably safer stimulating electrode that is very targeted to this precise area where we expect to see results, in fact, is able to have a behavioral effect in humans. So quickly, a thank you to all of my collaborators and to my funding. And much like Satyrus, I actually was planning to say that if you're a new student and you're interested in pursuing stimulation or even just recording on that single cell level in humans, we have a lot of exciting projects going on in our lab and we're actively recruiting. So get in touch with me or with Dr. Frieden. Thanks.